Okay, so let's take a look at mitosis. So mitosis, right? We like to call it mitosis. Why do we call it mitosis? Well, because it results in a 2N cell. So it's a 2N cell giving me 2N cells. They're just cloning the cells, essentially. So why would you want to make identical cells? Well, if you want to grow, if you want to replace cells that were dead or damaged, you got cut or something like that, your body must constantly make new cells. It's always doing this. Body cells are called autosomal cells or somatic cells, body cells. Autosomal because they rely on the autosomal chromosomes. Mitosis makes two identical cells. That's cloning. Asexual reproduction uses mitosis. We don't do that as humans, though. And 2N to 2N. Yeah, diploid to two identical diploid cells. So what do we have to know? Well, we don't actually need to know the phases like in biology 30, prophase, metaphase, anaphase, telophase, but it could still help. So the first phase is going to be the preparation. So we're going to prepare. We're going to make a bunch of enzymes to make everything happen, to regulate all these processes, to help the reactions occur. We have to get rid of the nucleus, stuff like that. The next one, metaphase. Okay, metaphase, the chromosomes are going to meet in the middle. Now we're talking about chromosomes here. And A for pull apart. So they pull apart. The chromosomes are going to pull apart. Okay, and then T for the end. Now you're lucky because you don't actually need to know prophase, metaphase, anaphase, and telophase, but if you're taking Bio 30, that could help. Prophase, metaphase, anaphase, telophase, pee mat. I think of a dog peeing on a mat. I don't know why. And I'm getting a puppy tomorrow, so kind of relevant to me. All right, so we do have this nice little picture here on the left, and I'm going to draw it on the right. So let's take a look at what's going on here. So first of all, we have to have DNA replication. So we're going to have our chromosomes. We're going to have, let's do this. We're going to have a pair of chromosomes. So we got a blue chromosome. And let's do a red chromosome for fun. They should be the same size. So there's a homologous pair. Technically, we could have another one, right? Just for fun. Anyway, so we have pairs. If we have pairs, that means it's 2N. So we have pairs. Therefore, it's diploid. And you know what's really annoying is actually, I got ahead of myself, I'm sorry. These are duplicated chromosomes, so they actually are down there. So what you would have before that is actually unduplicated, so you'd have something like this. A small one, big, small. So you'd have unduplicated chromosomes. Okay, and then how do you go from unduplicated to duplicated? You would have DNA replication. Remember, to count chromosomes, you actually just count these little centromeres, these little black dots here in this. So we have 1, 2, 3, 4. 1, 2, 3, 4. So we just duplicated or replicated this DNA. These are still 4 chromosomes. These are 4 chromosomes. So that's DNA replication. Now we're going to rip these apart, right? So now we're going to do mitosis. So now mitosis can occur. So the first thing you have to do is you have to kind of line them up. You know when you get Halloween candy? Here's all the Halloween candy. You got to put them all in the middle of the room so then you can separate them with your sibling and like one peanut butter cup to the right, one peanut butter cup to the sister, one Rolo to the brother, one Rolo to the sister. You got to evenly separate them. I don't know. That's what I do as a parent, and then I eat all the odd ones. So anyway, let's do that. So we're gonna put all these in the middle. So we're gonna line them up. There we go, something like that. Now they actually would probably be lined up like this. something like that. They should be in the cell. There we go. They're going to line up in the middle. So that's the meeting in the middle. Okay. And what that is that's going to be metaphase. Okay, so this is metaphase, but whatever, you don't need to know that. So they just meet up in the middle. Now, why would they do that? Well, they do that because then they can be pulled apart evenly. There's these little these little centriole things there. Okay, and then they go like this. These are spindle fibers, these little fiber thingies I'm drawing, and they just pull on it. Okay, there we go. And then eventually you're going to split them. It looks like I drew that the wrong way, so there. Maybe that'll work better, like that. And now what we're going to have is we're going to have this little red one's like this. There's going to be a little red one going this way. There's going to be a big red one going like this, and a big red one going like this. There's going to be a big blue one like this, and a big blue one, little blue, little blue. Right, technically these would all have those little black dots, those centromeres. They're like little handles for the spindle fibers to attach to. Oop. And then they're all kind of getting pulled. I'm not very good at drawing those. Okay, technically we don't care in Science 30 too much about this, but they'd be like that little noodle looking things. That's where those come from. There you go. And that's going to be anaphase. They're pulling apart. Okay, they're separating. And then the cell has to divide. So the actual cell divides. Okay, and now if we look at them, we have a little red, big red, little blue, big blue. And you'll notice that these cells in the end look identical to the first cell. Okay, big, big, little, little, big, big, little, little. Only we have two of them. Two of each. Just like that. And that's mitosis. So the whole purpose of mitosis is to make identical cells. Two-end cell, two-end cells, but we have two of them. These are called daughter cells. They're identical to the parent cell. Mitosis. 2n pairs. That'd be 2n equals 4 there. 2n, 2n. Perfect. Hopefully that cleared some stuff up. I don't know. But you can also watch this video right here. And it's probably better, to be totally honest. But it's too late for that.